Thank you very much for that talk, Ron. That was excellent. And for the last speaker of this session, I'd like to welcome Julie Mares. Julie is a nutritional epidemiologist in the Department of Ophthalmology and Visual Sciences at the University of Wisconsin. Julie has been studying the relationship between nutrition and eye disease for 20 years and will be talking today on the epidemiology of relationships of lutein and diet serum and macular pigment to age-related macular degeneration.
Good morning. Thanks for the invitation to this conference. It's great to see so many familiar faces and some new ones too. Thanks for the invitation of the opportunity to look at the epidemiologic evidence for relations between lutein and age-related macular degeneration. I won't be covering specific aspects of other aspects of diet that are related to AMD, but I should point out that lutein is only one of many dietary aspects that might protect against the development of AMD, as has been alluded. Other studies have shown fairly consistently that very high levels of fat are associated with the condition, as well as low levels of antioxidants and, in some cases, low levels of zinc. So we'll focus on lutein and its relation in diet serum and macular pigment to macular degeneration. The first evidence that there might be a relationship is that although lutein and its structural isomer zeaxanthin comprise 25% of the total carotenoids in the diet and in the blood, and in many other tissues, they are the predominant carotenoids throughout the eye, and particularly in the macular area, where they comprise macular pigment, which we'll be talking a lot about. Now, studies in RPE cells and in non-human primates have given strong biologic evidence that these may protect against macular degeneration through their properties as an antioxidant, their ability to absorb blue light, and these can lower the inflammatory triggers that are thought to promote macular degeneration. And as Shobha talked about, it's also possible that they may act systemically in terms of protection using these two mechanisms. What epidemiologic evidence will contribute is that lutein may have a relationship to AMD which develops over a longer period of time than can be studied in cells and in animals, that is in humans, with a wide variety of risk factors, and give some indication of the possible magnitude of this relationship. As the previous two speakers, while we of course are interested in whether lutein is related to the development or progression of advanced AMD, I really also agree that the money is on understanding whether lutein is related to the development of early stages, because if we can reduce the chances of developing early stages, the chance that people will develop AMD in their lifetimes is significantly reduced, and there's lower cost to society and individuals as well. Today we're going to be talking about lutein in three different exposures: lutein in the diet, in the blood, and in the eye. And they all have their strengths and weaknesses. We'll talk a lot about dietary lutein levels, which have the advantage in the results that we get from studies being easily translatable into recommendations for public health, such as what level of intake results in a one-fold, two-fold, three-fold change in odds for the disease. The disadvantage is when you ask people about their diet, you're obviously incorporating errors with regard to their memory of intake, and databases of lutein composition might contribute some noise as well. So many people like to look at blood levels of lutein in relation to macular degeneration. While it gives a shorter term estimate than dietary estimates. An advantage is that it also reflects the factors that influence absorption. But a disadvantage is that blood levels of anything reflect the internal milieu, which can be affected by disease states and many other things. So it's not necessarily a better indicator of status. We're very fortunate to have the technology to develop to look at tissue levels of lutein with macular pigment density estimates. We'll be talking about this as well today. And this is because, as we'll talk about, there appear to be a lot of influences on the ability to get lutein into the eye. And so the advantage is we can understand status of lutein specifically in this eye tissue in relation to AMD risk. First, I'm going to start with serum. This summarizes the data which describe odds ratios for both early and late maculopathy among people who have high versus low levels in the lutein in serum. Usually this is high as in the highest 20% and low in the lowest 20%. And I give the data in terms of odds ratios, the dots, and 95% confidence intervals, as I'll do on several slides subsequently. You can see here is the data point where in 1994, Joanna Seddon published in the results of the eye disease case control study that showed that there was a threefold lowering of risk for neovascular and exudative macular degeneration associated with having high serum levels of lutein. And similar results have been shown in a small study in France, but otherwise, the associations across study samples across different countries are really quite inconsistent, despite the fact that there's thought to be little noise in determination of lutein for serum, say relative to the diet. Now I'm going to show you a couple of slides that show you the relations for diet. I'm going to start with advanced AMD and then move on to early AMD. And over the last couple of years, there has been a convergence of evidence which suggests consistently that higher dietary intake of lutein and zeaxanthin is associated with lower risk for advanced AMD, and almost a twofold lowering of risk associated with usually being in the highest 20% versus lowest 20% in the sample. And this is fairly consistent, although not always statistically significant. And this is when limiting studies to those which have at least 50 cases and the most recently reported data for each sample. Sometimes in earlier samples, the association wasn't found. But you can see the picture for early AMD is quite different. There's a lot of inconsistency, where you see some associations between early AMD, between lutein intake and early AMD, but often not, and sometimes in the opposite direction. So what I'd now like to focus the next several slides on why the confusion. I think this will shed some light about confusion for a lot of risk factors for macular degeneration at the earlier stages. Now it might be real. It might be that lutein only protects against developing later stages of AMD. And currently the ARIDS trial is testing this, whether supplementation with lutein with and without DHA does slow the progression of advanced stages. So we'll find out about this in a couple of years. And so it may be that lutein really doesn't slow the development of early AMD, but it could also be that the observations are not real. And let's consider this possibility. It could be that the diet measures that we relate to AMD are closer in the period of time to when develops late AMD than to the period of time when one develops early AMD in these studies. And we change what we eat. Often we change what we eat in response to having cardiovascular disease or hypertension or things like this, which are also related to this condition. It's common. And as Ron has alluded to, we are concerned about selective mortality biases. We've tried to get a handle on some of this in the carotenoids and age-related eye disease study, which we've done. It's an ancillary study to the Women's Health Initiative conducted at 3 different sites in the U.S. They had, whoops, let's see. You told me what to do, and I'm thinking. Yeah, I did press.
This guy. Yeah, he's pressed it. It's not working.
So I'll tell you about the Karen study while this is being worked out. So the Women's Health Initiative had already collected dietary data on women in the Women's Health Initiative, as well as blood data in the mid-90s. And so we were able to look at the dietary data and select people who had the highest intake of lutein above the 78th percentile and the lowest intake below the 28th percentile. And we did this while we had about 2,000 people in this study. We did this for the reason of increasing the statistical power so that our study of 2,000 would have the power to detect an association, if there was one, similar to populations in the general population of the size of about 5,000. So with this dietary information, and also having serum levels, and Liz Johnson analyzed the serum for carotenoids and tocopherols in the blood. 6 years later, on average, we invited women back to take fundus photographs as well as to measure macular pigment density. We also collected more information about diet in different time periods so we could understand diet stability, and we also asked information about supplement use. And let's see, what was I going to say next?
Sorry, Julie, do you have your presentation on a memory stick?
I do.
Yeah, I'll take
So we can keep this sweep up.
Apologies for the computer malfunction.
I carry my notes with me always, thinking this might happen, but I've never used them. And this is actually the first time I haven't brought my notes with me. So I'm dependent on the mental cues.
Can I ask a question?
Yes. Yeah, Liz.
So, Julie, you talked about how you have this inconsistency among studies in terms of intake and risk. How much of a factor do you think is the method of assessment? Do they vary much among these studies as far as how they assess the tool?
I don't think there's a lot of variation in being able to classify somebody as having a whole bunch of lutein in their diet and little. So a lot is made about the noise that dietary intakes contribute to our understanding of epidemiologic relations. But it does a fairly decent job at classifying people at very high and low. And so I haven't seen any consistency with regard to the methods. Usually a food frequency questionnaire is used. Generally, you can identify people who eat boatloads of fruits and vegetables and very few. Where it's murkier is in the middle, which is why we didn't focus. There's more measurement error in middle intake. So I don't think across studies, especially when people look at the extremes in intake, there is a lot of difference. And I think what's interesting to me too is that there is very consistent data when you look at relations within a sample between diet, lutein, and AMD, and serum lutein and AMD. In the Karen study, we had very consistent relations, as did the eye disease case control study. So I don't think the methods matter a whole lot. And as we'll talk about with macular pigment density, I don't think the methods matter a whole lot in terms of relations of macular pigment density to to AMD. What I think is it strengthens the evidence to have a variety of different ways of looking at things to see if they all point in the same direction.
Mm-hmm. Okay, we're back. Now, Julie, maybe you could select your presentation.
Yeah.
Which folder?
Okay, talks.
Docs.
Cambridge. Final. Thank you.
Great. Just a second.
2 seconds, I should.
Mm-hmm.
Okay, so
Good.
Go all the way up there.
all right.
So you get a review. Okay, so this was the setup for the Carotenoid study, and now I'll show you the data. And this shows you the relationships between dietary lutein and early age-related macular degeneration 6 years later. You can see we found initially no association, like some other studies have found. But we had dietary information from several time points. We were able to look at women who had very unstable diets and exclude the grossly unstable, women with grossly unstable diets. And we saw that the associations moved in the protective direction, but were not significant. But we did see a significant age interaction, whereby we had significantly lower odds for early AMD among women under 75 years of age, and an association in the opposite direction in women older than this. Even though this was only 440 women, the power in this data set to change the associations was large, because there are a high proportion of these women who have early AMD. No, it might be busy. Okay, good. And in epidemiology, as Ron alluded to, sometimes you see an association and you think, well, this could just be chance, because you look at so many. But previously, I've seen differences in associations of diet to AMD in the Beaver Dam population when I looked at different time points. And also, I saw this also in a representative sample of the U.S. population, and Hayes too, in regard to also late AMD and pigmentary abnormalities, where you look at the earliest group at risk for the condition and see stronger associations with diet than you do among people that are the oldest in the group. We saw this with pigmentary abnormalities and overall early AMD and Haynes 3 as well, and in a population that was much younger, the atherosclerosis risk and community study, we see a strongly protective association. So I was starting to think by this time that this might be real. And if you think about it, the proportion of people in any study sample over age 75 that have early AMD is large. Generally, more than 30% of populations samples over 75 have AMD. We're about 15% under that. So it's influential in a data set. And so we were pondering this and went back to the literature, the epidemiologic literature that deals with studies of aging, and found that there is often a reduction in the power of risk factors as populations age. This shows a hypothetical situation for blood cholesterol, where you see a strong association among young people, and it gets weaker as people get older. Which is why the cardiovascular disease area has focused, especially in the early studies, on people who are the youngest at risk, to understand the more causal associations unaffected by bias. If you have a situation where you actually change your risk factor, as may be the case with diet or treatment for a particular disease, you can see a reversal in associations. So this is a possible explanation of what's going on with lutein and perhaps other risk factors as well. And this is further reinforced that it's not related to the lutein, but to something else by this slide, which shows odds ratios for early AMD among people in high versus low quintiles in 12 different age groups. Young age groups and older age groups. And you see this consistently among several different groups. We also see this consistently if we're looking at overall healthy eating index score or even serum vitamin D. So we think that it's something more general like survival bias and change in risk factor status. So it appears that high lutein intake is related to lower odds for advanced AMD. The data are unclear for early AMD unless you look at the youngest group at risk when it becomes more protective. So that strengthens the possibility there is some protection there. But in both cases, we have to worry about what that association is telling us. Is it lutein per se? Do people who have diets that are high in lutein have other habits that might be explaining these associations? And we've recently found that overall healthy diet patterns are associated with about a twofold lower of risk. And also that physical activity, as Ron talked about, is associated with lower odds for early AMD, probably related to the fact that physical activity lowers oxidative stress and inflammation if we don't do too much. And we also showed in two separate populations having poor vitamin D status is associated with higher risk for AMD. So all of these could be modified, could be related to the findings that we have here. They do slightly attenuate the associations that we see in this sample, but I think because of residual confounding, you can only remove the effect if you measure things perfectly. I think we still need to consider that some of the association we're seeing in epidemiologic studies might be explained by these other factors. And I think we need to have larger studies, which we can only do by pooling existing data, so we can help to sort out these individual factors that it might be reflecting. Of course, if you aren't certain about which of these aspects of diet are influencing relations to AMD, it's wonderful to have tissue levels. And we thank Billy Wooten and Max Snodderly for developing the techniques that we use in our studies. This minimizes the confounding by other dietary factors and also accounts for the ability to take up lutein and zeaxanthin in the eye. And I first point out that macular pigment density, using this method, does increase as dietary intake of lutein and zeaxanthin increase, but look at the amount of scatter. There's a lot of scatter, so that dietary intake explains only 3% of the variation in our studies in macular pigment density. You might think, well, maybe that's because you're not measuring macular pigment density well. It's so difficult, some people say, to measure using heterochromatic flicker photometry methods because you don't feel very exact about it when you're the subject. But indeed, we have quite repeatable data from time point to time point over 2 weeks using this particular technique. So the scatter isn't caused by the macular pigment density protocol. I think one thing that can contribute to the scatter is the fact that we all absorb different amounts in our diet and take up different amounts in the eye. Many people have reported that in the range of 25 to 50% of people who are supplemented with lutein have no retinal response. And this is using many different methods for measuring macular pigment. Also, in the CAROT study, we have found evidence for dietary influences on the ability to accumulate retinal carotenoids. For example, we find that macular pigment density is lowest among women in this study that have a low intake of polyunsaturated fat. after adjusting for the other predictors of macular pigment. Also, and I wanted to say that this is consistent with other evidence which shows us that some fat seems to be needed for optimal absorption. Having salad dressings with fat increases absorption of many carotenoids. Plasma lutein response is greater when lutein's supplied with a high fat spread. Studies have shown that eggs, which have a small amount of lutein but a large amount of fat, do enhance macular pigment density beyond what you would expect from the small amount. And Liz Johnson also observed a trend toward foveal enhancement of macular pigment with DHA supplementation in a particular study. So I think the possibility that fat does modify the ability to take up lutein is strong. Also, we found that after adjusting for dietary lutein and zeaxanthin, that women whose diets were higher in fiber had higher macular pigment density. Why would this be? Well, fiber, I think, could simply be a marker for the intake of whole fruits and vegetables and whole grains, which contain not only the antioxidants, which we think about often, like vitamin C and vitamin E, but the myriad of phytochemicals, polyphenols and such, which can be antioxidants. So it could be that it's a marker for antioxidants. Higher overall intake of antioxidants would slow the turnover of other antioxidants, sparing them for the tissues. So this is a possible explanation. Also, we found that waist circumference predicted macular pigment density, as I've shown you before. So at all levels of dietary lutein and zeaxanthin intake, women who were lean had higher macular pigment than those who were obese. Also, we found women who had diabetes had lower macular pigment compared to those who did not at all levels of dietary lutein intake. We've been pondering why this is for some time. Others of you have found these associations. And Liz told us that abdominal adiposity that she gets in the lab is often yellow. We've been thinking about why is that and what might be explaining this association, and wondered if possibly there was an evolutionary advantage because the abdominal adipose tissue is a source for breast milk, so it might be a competition between two different pools in these women. It might also reflect the fact that obesity and diabetes are associated with higher oxidative stress and inflammation, which could increase the turnover. Or these might be markers for other host factors that influence the uptake or transport of carotenoids. Such as other host factors regarding HDL level or genetic markers for HDL, as we'll talk a little bit about at the end. Or it might be markers for having metabolic syndrome or genetic markers for metabolic syndrome, such as CD36. So overall, we had a wealth of information on diet and lifestyle and health in this sample, but we were only able to explain 12% of the variation in macular pigment density based on the markers that we had. A small amount in diet and a small amount from these other sources I've told you about. So something else is influencing macular pigment density, and the twin study that was published about 5 years ago suggested that 70 to 80% of the variation in macular pigment may in fact be explained by genetic factors. And many have proposed these factors. Paul Bernstein, I'm sure, will have a lot to tell us about candidates related to xanthophyll binding, which he has worked out beautifully in his lab. So we've considered that these might be some of the predictors of genetic risk or genetic determinants of macular pigment. Also, there's a strong evidence to suggest that variants in some genes that influence the transport or the stability of HDLs could be related to carotenoid distribution, such as is evidence in people who have Tanger's disease or particular chickens who have mutant ABCA1 who despite high levels in the diet are unable to maintain serum levels and levels in most tissues of carotenoids. There are several other gene variants that are related to HDL stability or transport. Recently, the Boral group has found that CD36, which is related to HDL level, is related to macular pigment density. And ApoE, which can affect HDL level as well, has been related to ApoE in this UK group itself. There are others that haven't been studied. Also, we know that lutein absorption is related to the variants related to SCAR-B1 in intestinal cells and in the retina as well. And then there's recent evidence for a relationship between the enzyme which cleaves beta-carotene to form two retinal molecules in relation to macular pigment, and we don't really understand the basis for this as well. We'll be further studying this in carids, as well as whether levels of lutein in the diet, serum, and macula are more strongly or less strongly protective in people who have higher risk for other genetic markers for macular degeneration. Finally, because there are all these unknown, unpredictable factors, it's best to measure macular pigment directly and study it in relation to status of lutein and zeaxanthin, as Richard and John have done in autopsy specimens. I expect they'll tell you more about this study, but about 10 years ago, they really increased our interest in the possibility that macular pigment might be related to AMD. And so we've been looking in people, along with many other people. And you can see this is the evidence that exists today. So several different studies using different methods for measuring macular pigment in samples that are usually cross-sectional, where macular pigment and AMD is assessed at the same point in time, although in one recent report, longitudinal. And then studies which vary in size between about 100 and 1,700 people. You see that the data are mixed. Some studies finding an association, 3 of them finding it, and 3 of them not. In the case of the Bernstein paper, they found the association only when they removed people who were taking lutein supplements, and this makes sense because if you're taking lutein supplements, it does increase macular pigment density, at least in some. So that would, if the intake was recent, that would make sense. In the one longitudinal study that was done, an association was found, but it was not statistically significant. And in the carids population, I'll show you the data next, we found that we do see it, but again, only in people of younger ages. So this shows you the odds ratios for early macular degeneration among women in quintiles 2 through 5 for macular pigment density, who had a density of about 0.4, compared to those who had a density of about 0.1. You can see there was not a significant association. We looked at the whole sample. And we found a marginal association when we again looked at the youngest group at risk. But it was not statistically significant. And again, you see the association going the opposite direction when you look at people over 70. So again, this bias of older people does seem to be influencing how we interpret the data from epidemiologic studies. So I think we need prospective studies, and we need to study people as early as possible as we possibly can. As both Shobha and Ron alluded to, what we see on the horizon is having a better understanding of interactive effects and joint effects. People who have one risk factor, is lutein more important if you have hypertension or obesity or diet's poor in other aspects? And really, our epidemiologic studies and single studies have not been well powered to look at interactions. And there is a lot of preliminary evidence for the possibility of joint effects. Also, the whole paper that was just published last month showed that people with the high-risk complement factor H allele in those people, the protective effect of various dietary factors, antioxidants, and zinc was stronger than in people who didn't have that. So we're doing a lot of genetic work in many studies, and I think we'll be able to have some answers in the next couple of years. So in summary, the epidemiologic evidence for relations between lutein and AMD are consistent for late AMD. The magnitude is potentially strong, between 2- and 3-fold effect for people in the highest 20% to lowest. But the caution is this could reflect some aspects of healthy diet or healthy lifestyle, which we still need to tease out in larger studies. The data are inconsistent for early AMD, but I think we have enough evidence to indicate that this might reflect lower susceptibility in the oldest people studied, and bias relative to diet change and survivorship. And certainly there's evidence increasing that the protective relationships could be influenced by variable responses to dietary lutein, to other aspects of diet and health which influence lutein intake and absorption, such as dietary fat, dietary antioxidants, as well as obviously genetics and joint interactions with many of these together. And I do want to acknowledge the many people who contributed to the data that I showed today. I'd mentioned many of whom that are here today. I mentioned Liz Johnson, who done the serum analysis, and Billy Wooten, who together with Max developed the macular pigment density techniques that we used in our study. I think all epidemiologic studies stand on the shoulders of what Barbara and Ron have done in Beaver Dam and elsewhere in terms of helping us have a means for detecting early as possible maculopathy photographically, which provides enormous insight. And they also provided the photographic grading for many of the samples that are reported here. Thank you. Do you have any questions?